Julie Harless, Professor of Biology today. Julie's made numerous presentations in the past. Thank you for putting all this together for us. And Julie's title is 2015 Nobel Prize in Chemistry, DNA Repair and Why You Should Care.
Okay, so I cared deeply. I got all excited when I heard who won these Nobel Prizes because this is a field that I did my graduate work in and after, postdoc. And so when I heard the names, I knew who they were, and it's like, oh my goodness, because we used to laugh. Nobody's ever going to get the Nobel Prize in DNA repair, right? Wow, they did. This is cool. So I thought I'd give you a little bit of background on what we knew about DNA repair and when. So to put their work in context. And then I do a little quick overview of how exactly what each one of these three guys did, because they did really different things. And then we can talk about the kind of implications for when it doesn't work. That's always how we know that something exists in biology. We're just happily going along, there's not a problem until you find somebody that has a mutation and they're not doing something they should. And it's like, oh, so there's a mechanism for actually doing that. And so DNA repair was another one of those things. Obviously, we didn't know a lot about it until we knew about DNA and that was the genetic material. So we're talking right after World War II. Good evidence for that. There had been some earlier studies looking at DNA that did finally prove that it was the genetic material. And along with some of those studies, they were able to do these inactivation experiments where they would take the DNA and irradiate it with ultraviolet light. And what they would see is it didn't do the transformation that would normally happen, that one of these the bacteria would take it up and the new genes would be expressed, and they weren't if you irradiated the DNA first. But then they found out if you hit it you do UV irradiation to do some sort of damage to it, and then you hit it with bright visible light, that it would regenerate it. And it's like, okay, well that's kind of weird, but until we knew what DNA was, they weren't able to explain that. And once they knew what the structure of DNA was, it's like, okay, we know that this is a stable molecule because you have to hand it down to your grandchildren. No experiments, but just theory, right? Are we coming? Oh, good. How many? How many chairs do we need? Okay, I'll stop here. That's okay. Let me squeeze it.
It could be chummy.
I don't mind sitting next to dinner. You guys can hold on this phone. Somebody could be playing this very candy baby back. There's room over here.
And we can do a double row over here. Hi.
As usual, disruptive biotech. Sorry guys, appreciate that.
These two chairs aren't taken either, so.
Keep going.
And then coming and coming.
That's fine.
There's two empty chairs here. Yeah.
chair back there, please. Thank you. Somebody can sit right there.
Yes.
Got one here?
Sit here.
All right.
Yeah. Yeah.
Thanks, guys.
Thanks for coming. So I was giving a real quick overview of kind of the history of how we know that DNA repair occurs. And so we had these observations before we really understood what DNA structure was. And then once we knew what the structure was, there was this assumption that this had to be a very stable molecule, because you hand it down to your children and they hand it down to your grandchildren. And so it was an assumption that this was stable, unless there was some sort of insult. So either physical insults like radiation or chemical, but that it stayed pretty much the same. The early stuff, the early examples of radiation damage was early 1960s. They showed that ultraviolet light, when you shone ultraviolet light on DNA, you got thymine dimers. And biochemistry was so far ahead of molecular biology, they were able to decide determine what a thymine dimer was. So that's when two thymines are next to each other in the DNA, you irradiate them, they absorb the energy from the ultraviolet light, and they bond to each other rather than doing hydrogen bonding across the helix. And that starts causing some problems. They didn't realize what they were, but they were able to define them. A lot of the work on DNA repair then happened on these thymine dimers because they were easy to induce, because everybody's got an ultraviolet light, right? Or they were cheap enough to buy in the lab. And they're chemically really stable. Later, if you start doing changes to DNA bases in other parts, you start making an unstable compound, and then they're very hard to study. So ultraviolet light thymine dimers were the gold standard, what everybody studied, just because you could put a known number of dimers in a piece of DNA by using a particular dose, and they weren't going to go away. You could put it in the fridge, you can put it in the freezer, and we can come back and study it later. Later, I studied some lesions that were horrible because overnight, about 50% of them fell out of the DNA. And I had to work late nights running things through an HPLC and trying to purify them because they were so unstable. So dimers were wonderful, and they became the model for a lot of this DNA damage. So I took this from the website from the Nobel Prize Committee. So we've got 3 guys all working on slightly different aspects of DNA repair. So Thomas Lindahl is looking at base excision repair. So for those of you who are not familiar with the structure of DNA, we've got a backbone of sugar, phosphate, and then bases sticking off the side. So Lindahl is working on a repair mechanism that just takes out the base and it leaves the backbone intact, which was a little bit weird. That came later after we understood nucleotide excision repair, where there's damage and we're going to take out a whole strip of the backbone and fix the whole thing. And then Paul Modrich is looking at a different system in which there's mistakes made during the copying process when we're making new DNA before a cell divides. The enzyme that does that is DNA polymerase, and it makes a mistake here and there. And so the mismatch repair fixes those. The first 2 can be done pretty much any time during the life of the cell. This is going to be done just after DNA synthesis. Okay, and then I just wanted to show you what a small world this is. So the first paper there is one by Thomas Lindahl, and he cites one of my papers. Yes! And then there's some other people here that are writing about it, and they're citing us and Sankar. So it's a nice little group. Okay, so let's look at nucleotide excision repair. That's the one I know best because that's the system that I was looking at. So the simple version here is that we've got ultraviolet light has caused those two thymines to stick to each other rather than base pair across the helix. And normally the bases are off from each other by about 36 degrees. And they go around. So when you get them doing this, what it does is it puts a kink in the backbone. And that's the secret for the way excision repair works, is we've got this series of proteins, and this is in E. coli, UVR, A, B, and C, that will recognize there's something wrong with the backbone. They're not looking in here, they're just seeing that there's a distortion in the backbone. This extends for about 7 bases on one side and 8 bases on the other. So it's a slight little distortion. The angle's wrong essentially, but this protein binds so specifically, if the angle's wrong, it recognizes that. And when it does, it calls in the other components here, so you get this complex that cuts the DNA on either side. And so this little patch with the damage pulled out, and it gets filled back in by DNA polymerase 1, which is not the one that normally copies DNA, so it does a short patch repair, and then we're going to ligate it back. So we just cut out the damage. It turns out that works because it works on thymine dimers because they cause that distortion. It also works on the kind of damage that I was working on, where we chemically damage the bases by adding in a big group onto, and that messes up the spacing in there because it's really tight. And so anything that distorts the helix will set off UBR ABC repair, and you'll get that patch stuck in there. In humans, of course, it's not as easy as it is in E. coli. There's a disease that occurs when people can't repair ultraviolet light damage. And the proteins, it's kind of the same way. Of course, we've got a lot more proteins involved, so I'm not going to do the details on that. But the disease is called xeroderma pigmentosum. XP for short, for a reason. What happens is that the people get skin cancers. So all kinds of skin cancers, basal cell, some melanomas, just all kinds of skin cancers, starting at the age of 5 or 6. So that tells us, that's one of those signs, we're doing this all the time. Because if you can't do it, you get these horrible invasive skin cancers. Hi. There are some neurological problems also, which if you think about, well, ultraviolet light is not going to penetrate into the brain. That tells us probably some of the chemical damage. Oxidative damage that's causing damage to the bases is probably involved in the neurological damage, and then that builds up over time also. But usually they don't live long enough for that to be a big issue unless you diagnose it really early. It's a recessive disease. And there are so many different genes, usually it's really rare. But if you do have it, then and your family knows about it early enough, what they do is they keep the children inside during the daytime. They never go outside. It's a very disruptive of family life and no cure, obviously, because it's a genetic disease. So the genes are called XP genes. So there's XP A through G, I think. Most of them, if you are defective in XPA and defective in XPC, one copy of A is good, one copy is bad, one copy of C is good, one copy is bad. They will complement each other and you'll be normal. So they're independent genes. There are a few of them that don't complement, but that's why, because there's so many genes, there's very few people that are going to have a defect in both copies of their XPA. So it's a very rare disease. Okay, so that base nucleotide excision. So we take out that whole string of nucleotides and replace them. So that was Sankar's work. Thomas Lindahl's work, he's looking at a defective base and getting that taken out. And his work was interesting because he worked on that photolyase that will do the direct reversal of dimers during his graduate work, and then went away and did something completely different as a postdoc. And at one point he was doing some calculations and of the chemistry and this assumption. So he's coming into this in the mid-1970s, early 1970s. And so people had been looking at thymine dimers and things like that for 10 years or so. And so he starts thinking about chemical damage and how stable is DNA really? And he was more of a chemist than most of the biologists at the time. And from his calculations, he could see that the amine group on cytosine is not terribly stable. And at at body temperature and neutral pH, cytosine should deaminate. It should fall off at some given rate. And he calculated the rate. And then starts multiplying that by the amount of DNA in our cells. And it looks like we should have about 1,000 deaminations a day. Total in our cells. So we're talking something like 10 to the 13th deaminations over your lifetime. The odds are that's one of them is going to be in a bad place. And when you deaminate cytosine, it looks like uracil. So now it's going to base pair with thymine rather than with guanine. Or adenine, sorry. Adenine rather than guanine. And so that's going to cause a lot of mutations over time. And why doesn't that happen? And so when he's looking at it, what he's seeing is that there is a mechanism and there are enzymes. So he starts looking for enzymes that will recognize when there's a problem with a base, cut out the base. And now you've got this weird thing that is DNA, intact backbone. So our sugar phosphate is all intact and we're missing our base. So they call it an AP site, an apurinic site, because it's missing a purine. Turns out that is really easy to break. A little bit of base, a little bit of acid, and it pops open, and you get a break in the backbone. And that is the basis of the repair process. So that broken piece of DNA is then a recognition site for enzymes that will chew it away and replace it. And it looks like in two different kinds. You can get short patch and long patch repair, which is how many nucleotides get chewed away. But we can replace it. So in some ways, it's sort of like the excision repair at the beginning, but the first thing is taking out that base And people didn't think that that happened for a while, but it did. Okay? So that's a different set of chemical problems. So nucleotide excision repair will find big things that disrupt the base pairing in the helix. And base excision repair is going to recognize smaller issues that don't interfere with the base pairing, are not going to disrupt the helix, but they recognize that that's a base that doesn't belong there, that there's some problem. Okay. And then we have the mismatch repair. So this is the Modron's work that occurs right after DNA has been copied. And so what we've got up here, we've got strands of DNA, and we've got a problem right there, right? T doesn't bind with T. So it doesn't match. And the polymerase is kind of measuring the diameter of the helix, that new helix. It should be all the same, is it 10 angstroms? Yeah, it's 10 angstroms wide. If it's less than that or bigger than that, this enzyme's going to recognize that we've got a problem. The base pairing's not perfect. Now the question is, we got T's on both sides. Which is the good one and which is the bad one? And you could just randomly do that, right? Fix that one way or the other. And so the beauty of what he's found is that we're using the methylation. So DNA is methylated at certain sites, right? Are they restriction-looking sites? Are they palindromes? Methylation sites?
No, I don't think so.
Is it sequence specific?
No, I don't think it is.
Just randomly methylated? Okay. So as DNA ripens, after matures, after it's been copied, it will get methyl groups added on to guanine? Adenine. These people have taught genetics since I have. It's usually on one, wait, it's going to tell us here.
CPGI ones, but I'm trying to remember the...
Probably C's, yeah. Yeah, so we're going to have a methyl group added on, and it, so that's going to tell us that's the old strand. It's been around longer. The new strand will not be methylated yet. And so we've got to have kind of again, a complex of proteins. One of them recognizes the mispairing, that the sequences are not base pairing like they should. And then we've got to have another one that goes hunting down the strands, looking around in this region to find a methylated site. So it doesn't have to be really close, sort of close. And binds to that methylated site, and then is able to, we got another protein here that goes, okay, this C is methylated. Oh no, it's the adenine. Look at that. Okay, so the adenine is methylated here, so this is the new strand. So we're going to cut that strand out and fix that one, not that one. Right? So we've got to be able to tell which one is right. Yes?
Are questions allowed?
Yes.
I had a question regarding the repairing mechanism of the genome. Does the DNA has some kind of aging process which affects its mechanism of repairing related to the hidden mutations?
I think any of the adducts, that the mutations that are there, if it's around long enough for that second strand to get methylated, then we wouldn't be able to tell which was the good strand and which was the bad strand.
You know, statistically, people when they get into aging, coming to the aging process, the chance that they get cancers or some kind of different problems.
Right, because they accumulate. Yeah. So if so, what they have seen is like if you're out in the sun a lot, There is kind of a window of how long the UV repair mechanisms will work. So if the dose is under a certain level, then most of them are going to get fixed. If the dose is higher than that, there's only not very many copies of the DNA repair proteins in a cell, and they're not going to be able to fix all the damage. Once the DNA has been copied, sometimes the polymerase will just push through. It's like, I can't read that, but whatever, and it sticks something in. If it is in heterochromic regions, so the DNA gets wound up with the histones, then it's not going to, that region of DNA is not going to be available to the repair proteins. So some sites are not going to be repaired as well as others. Some of the work that was my dissertation showed that genes that are being expressed are more likely to be repaired because they're unwound and they're not in the heterochromatin. Because that heterochromatin is going to just exclude the proteins that have to come along and monitor where the damage is. So we're going to talk a little bit about the aging process too at the end of this, okay? And now I've forgotten what this link is. Shall we find out? Oh, okay. This is a site at the Nobel Prize Committee site that kind of goes over this. No, never mind. That had better pictures. This makes sense? Do you guys have questions about this? Good with that? Okay. So this is a picture of a little girl with xeroderma pigmentosum. And I think she's something like 6 or 7 years old. And so some of these are going to be malignant melanomas. All the rest of these are also skin cancers. So it's no repair is a problem. So did you know every time you go outside in the sun, you're fixing a lot so that that doesn't happen to you. There are, again, the neurological abnormalities also with base excision repair. So this is the one that takes out those, just the base that's occurring 1,000 times a day. Okay, what they found is, again, it's a recessive disease. So if you don't have any enzymes that are able to do that, you're not born. Because just during the embryonic period, you accumulate enough DNA damage that it kills you before you're born. So most of the time when we see problems with that, it's because the mutations have occurred later in life, that you're not born with them genetically. And so we'll see things like when you start seeing mutations in P53. Once you get those sort of mutations, you're more likely to accumulate more mutations. And it looks as if, and statistically, that the DNA repair enzymes are more likely to be mutated than other genes in a cancer, which then means you'll accumulate even more mutations. So for some reason, those look like mutational hotspots. And then if you can't repair damage, then you're going to get more damage and more and more and more mutations. And so that's why cancer seems to kind of run away. So the initial mutation, at least for this one, is probably not a genetic one, but you'll see they've seen patients that had a mutation in P53 that also could not do excision-based repair, and they found that sort of combination in sarcomas and breast cancers and brain cancers and adrenal cord. So it's not specific, it's kind of generalized. That when things go bad, they go really bad. The mismatch repair, they found the genes that are responsible for that mutated in all kinds of cancers. And this one also seems to turn off So when there's a mutation, when we're not doing mismatch repair, it turns off the other DNA repair genes too. So again, we get this accumulation that it kind of feeds on itself. So one question would be, why are these genes more vulnerable? And it could be just that they're expressed all the time, so that the way the DNA is oriented in the nucleus, it doesn't have the histones wrapped tightly, and so it's easier for the radiation, easier for the chemicals to get there, and these are going to be genes that are expressed at a low level all the time. So they're likely to not be in heterochromatin, that they're likely to be in the loose chromatin. But there's help. There's an essential oil that will turn on your DNA repair systems. There are several of them. I mean, there are all kinds of ways to save your skin. That you just put this on and it'll beef up your repair. My favorite, this is so cool. Oh no, there's no sound.
I work with you all the time. Dr. Harless, I work with you all the time. Are you being sarcastic? Or
Just a tad. And this is just so bad because this whole thing, you can subscribe to these weekly videos and it has this sound that's apparently just the right wavelength that is going of sound that is going to repair your DNA. And I think you can meditate to it, yeah. Okay, so I'm guessing that a whole lot of those skin creams and the essential oil and probably this have hit the internet since it was announced that the Nobel Prize was for DNA repair, right? Every time you hear something that has to do with health, somebody's going to try and make a buck off of it. So yeah, so DNA repair is the buzzword now. No, you can't fix it from the outside. Yeah. Okay, so one of the things that looks a little bit intriguing is that, okay, we've got cancers that are not doing DNA repair like they should. Well, can we actually beef that up so that the cancer cells start accumulating even more mutations, which starts acting like chemotherapy? Because the original chemotherapies, those are nasty drugs. They cause damage to DNA. And the idea is that growing cells, which would be tumor cells, tend to be growing more than your other cells. You give those drugs, they're taken up by the growing cells. They cause a lot of DNA damage to the point that the DNA doesn't work and it kills the cells. So can we use the same thing for the DNA repair? If they are not doing DNA repair, can we augment that so that they accumulate enough damage that they set off their own suicide program, or that they don't function anymore? And I think that's Mark Kelly, which is, I used to work with him.
So how did they, because your skin cells are rapidly dividing as well.
Yes.
Did it mention in that paper how they target the cancer cells?
No, I think this is mostly just theory so far. I'm not sure.
It's a great idea, though.
It's interesting. I'm not sure how to implement it without causing, because I mean, all the trouble with chemo was that you cause so much damage to not just the tumor, but to your bone marrow and to your skin and to your gut and to your hair. And so targeting is again a problem. I mean, it's not like you want to turn off the repair for everything, because then you're just going to cause more tumors. So controlling that one.
Good job, Christopher. No, I'm just sorry.
Okay. Okay. Yeah, CRISPR's going to fix everything, isn't it? Okay, so that's kind of my overview. I was hoping you guys would have questions or comments or exciting ideas about where we go from here. Yes.
Just one follow-up question.
Yes.
Did you say in the beginning
I might have.
that, so you have UV light, higher energy light, gets in, you go out in the sun, and
You get the
damages your cells, damages your DNA. And then did you say
Visible light.
you go back into visible light and we see a repair that occurs?
Only if there is a protein there that.
Of course, protein is the best.
Yes, yeah, so it's, yeah, so that's, yeah, you can't go into just lower wavelength or lower energy light. So
is that
the
UV, or is so all the sunlight that we see is visible?
Yeah.
We see, right?
Uh-huh.
Does that light activate that protein?
Yes. So the protein binds to the dimer, and then it is activated by the light, changes shape, and pops the dimer.
So it's good to be out in the sun with, like you said, as you said, with sunscreen, with UV protection, and then the visible light will.
It's not clear how active photolyase is the name of the
Oh, it is photolase.
is the name of the protein. How well it works in humans.
Is there a link to vitamin D?
Not that I know of, but the last people that I knew that were studying photolyase were in the 1970s. So it was studied in E. coli and all of those and bacteria. There was one woman who was looking at it in humans, but she was looking at cell extracts, not intact cells, and was getting a little bit of a signal, but there was some question about how useful that would be under real biological conditions. Because one of the problems is just the permeating the skin. The group that I worked with in Holland was a couple of different people were working on UV dimers, and they had this really gross assay. So we were making monoclonal antibodies to thymine dimers and other kinds of adducts. And so mine were some chemical adducts. And so there was a group that was doing dimers, and they had what looked like a really good enzyme, or a really good antibody. And what they would do would be they would get human skin, patches of it, from tummy tucks and breast reductions, mostly. And then you would tack it onto a board and you would irradiate them to different levels. So the cells, it's fresh, the cells are pretty much intact. And so you would give it enough radiation that they would get a little bit of redness, and they called them arithmetic units, so how red you got, so they could judge that. And then they would take a slice of the skin through the depth of the skin, put it on a slide, flood it with their antibodies, and see how far down did the radiation go. And human skin has about 8 layers of cells, and they would get down, and they could detect 2 or 3 dimers per cell with this antibody. It was a good antibody. And so you could see how far down it was. And so as long as you didn't get the damage, too much radiation, too much sunlight to get down to the stem cells, you were okay, because these guys are going to die anyway, right? And so they were able to see what kind of doses those were. And the guy who did it was always, asking for volunteers. And I kept trying to explain to him that genetically I didn't fall into the same group as the Dutch girls. And of course, the problem there was that when we're talking about tummy tucks and breast reductions, if you did that in this country, that would have been skin that was never exposed to sunlight. So it would be pretty pristine skin. In Holland, that's not true.
Understood.
Topless, I bought a bathing suit there that I had to explain to him would be illegal in this country because there was no top. So the rules are different.
Taking that out of my
I'm sorry, what a picture. It was a long time ago. They were not so saggy. Sunlight. He said he'd take that out, right? So.
That is on YouTube, baby.
So most of the damage that we're really worried about in humans is going to be that deep damage, and we're not going to get much visible light down there either.
Exactly.
So I'm not sure how good the photolysate would work for us.
I have a question.
Yes.
Are we at the stage now where we should all get genetic counseling, whether sick or not, so that we have a profile, and as these various modes of repair develop, we can take advantage of it, rather than waiting till it's too late?
I guess it depends on your outlook on life. I'm more fatalistic than that. It's like we're all seriously defective in a whole bunch of genes, and if you sit around and worry about it, you're going to ruin your life. So,
But kind of following up, if you knew that you could pass something on to someone else, you could make that choice, right? So maybe that's another way of looking at it.
yeah,
I'm
so
with you.
so you would, yeah,
Side of the coin.
yeah. I mean,
Well, for
it-
example, if there's a pattern of disease in the family.
yeah, then you would want to look at it. But I mean, what are the estimates now on the number of lethal mutations we all carry as?
Enough to be lethal.
Well, no, I'm thinking that are recessive.
Right, that's what.
Yeah, and so I guess I tend to be fatalistic about this because they keep talking about the cancer rates keep going up, and people are, more and more people dying of cancer, and that's because they are not dying of heart attacks younger, right? That you get cancers when you're older, so that's kind of a good sign. But nobody's gotten out of this alive. So
These websites that sell these profiles reliable, or is there a better place to go for such?
oh, like 23andMe, I think they're probably okay. I think, weren't they sued recently and shut down for a while because they weren't reliable enough to make healthcare decisions on them. So they could tell you where your grandparents or great-grandparents, what continent they came from. But they were also doing kind of genetic testing and reporting out disease possibilities. But no, their sequencing wasn't good enough for that. And the government shut them down. But they just let them open back up and continue to do that.
And they keep all that data. Just saying.
Yeah, and the problem is that they tell you that you have an increased risk for blah, blah, blah. They don't, but there's no genetic counseling that goes along with it. And it's like, people tend to think that if you tell me that genetically, it's like, okay, well, I'm doomed. I might as well not bother to eat right or exercise or quit smoking or do whatever because I have this genetic, mark. And
No, the 23andMe doesn't give you anything
Does
that's
it not?
that
I don't know.
interesting. No, I just
I haven't
haven't
been on
done
the site.
oh, none of them are interesting?
No, nothing that's going to
They just give you ancestry.
Well, no, that was because they were shut down because they were given.
Mutations, but when you go to the databases, the mutations that are meaningful mutations.
Oh.
Nothing that's going to indicate that you're going to come down with.
Okay, because I think they were doing that, and that's when they were told that their sequencing wasn't good enough to do that, and they didn't have and they weren't doing any genetic counseling. And I'd heard that they'd come back up, but maybe not the disease counseling part.
The only interesting thing they tell you is whether you're lactose intolerant or not.
It's like you sort of know that. Eat some ice cream, and it's a better way to find out.
It's going down, it is.
Yeah, unless you are, but
Two things. This is a cart beside the horse, rather in front of it.
Okay.
There are people making these decisions already based on limited genomic data.
Yeah.
So I had a student 2 semesters ago that chose for a double radical
Oh.
with no history of breast cancer, with no evidence of breast cancer, because she tested positive for glyphosate.
Yeah.
So that's the decisions that are being made now.
Yeah.
Now, it's a little young yet, but I think in another few years, sites like that will be user rated. I was talking to someone the other day that found Yelp reviews on fertility and abortion clinics.
Oh, man.
So
Okay, so if 23andMe is not giving me the information that I want, then they get a bad review. And so somebody should come up with a company that gives me, wow.
I think it's going to get there.
Ooh, that's scary.
It is. Absolutely.
Yeah.
Part of it too is we took some of the genomics students to Baylor on Friday, I think it was, and we were talking to the head of the cardiovascular molecular diagnostic lab, telling us all the latest and greatest. But Baylor College of Medicine is the leading genomics center in the world.
Right.
And so he's tied into that, and he's on the clinical side. But part of the problem is about the genetic testing is there really still is a paucity of data.
Right.
We have a lot of genome sequencing. So one thing that he was telling us about, and this has been in the news a little bit, is the Million Genome Project that has been out
Right,
there recently.
right.
The ability to sequence is, our students in here can tell us all about this now, but the technology for sequencing is progressing faster than Moore's Law. So it's actually more than doubling every 18 months in terms of how much we can sequence. And the price is dropping. So we're now, for the first time in history, in a position where we can even plan this million genomes. So it's going to be 1 million volunteers crossing all ethnic groups and genders. And that once we have 1 million genomes, the idea is we'll just have enough, we can run good statistics on some of these things and have better data. It's never going to be perfect, but it's a concerted effort that it's much aligned with what the Human Genome Project was
Right.
when it started back in the 90s. So, and in fact, it was interesting that he was saying that right now we're so far away from results from that we don't even have a building that can store the million genomes yet. So we have a building first to store them. So
It's for servers.
There's one in Utah. There's one in Utah.
Yeah.
Well, there you go.
Yeah.
And just a quick little clarification, I phoned a friend with the question that you stumped us all on earlier, and it's the A's are prokaryotics only.
Oh, okay.
A's so it is CPG islands, which is cytosine phosphate guanine, and about 70% of the CPG islands, which are all over our genomes, are methylated.
Is it the C or the G?
It's the C.
The C.
And they just upstream of the promoter region.
That's right.
Yes.
Yeah.
Okay.
Among other things.
So there is some sequence specificity that it's the islands, not just any C in a,
It's CG.
yeah, okay.
But it's not as specific as promoter
not
region.
valid.
Right, or restrictions.
And by the way, 23andMe on their website says that they'll give you carrier status reports for cystic fibrosis if you're a carrier for the cystic fibrosis, sickle cell anemia, or hereditary hearing loss mutations.
If you pay extra.
If you pay.
Do they have all the mutations for cystic fibrosis? There's a whole
I did not
bunch of
say anything
that.
like that to them.
So I wonder if they're actually sequencing or are they just using probes? Right, you can use probes for that. So if you know, it's just cystic fibrosis has so many mutations.
Well, they're sequencing because the mutations that they do give you information on, they give you your sequence.
Oh, okay, so they are sequencing. Okay. Yeah. Did this get really too technical for you guys? Or did it make sense?
It was wonky.
Okay. I mean, it's pretty nerdy. Yes.
Actually, I heard something on the radio this morning. I heard a snippet, and I'm hoping someone else may have heard it, and it's a little off.
That's fine.
topic, but not-
We're done.
Someone turned in results of a clinical trial, I believe it was, of some type of cancer. Like I said, I only heard a snippet, that they had 97% cure rate. Did
Yes,
anybody hear that?
leukemias.
Hear
There are
that?
certain leukemias.
It was a
And it
leukemia.
was,
You're right.
Myelogenous leukemia.
what's the word?
Yeah.
Auto, I mean, I'm losing the word, they take your own cells.
Oh.
Right, it was.
Okay, so yeah, so they're programming your immune system to go after your tumor. I saw that too. I was reading about it, and they were saying that it works for blood tumors, but they can't imagine how it's going to work for solid tumors. But they had gone in, they'd taken the T cells out and reprogrammed the receptor so that it recognized only the myelogenous leukemias, so some antigen on them. So just really targeted it. So they must have, they found a monoclonal that had a good receptor, and they go in, I'm guessing using CRISPR, and replace that in your T cells. And so now instead of having T cells that recognize different parts of the receptor all over the place, all of the T cells are going to recognize that one epitope and go after it. So it makes it really specific. And they did have enormous, I mean, there were just one or two people it didn't work at all, but for most of the people it was a complete remission.
Well, I don't remember the numbers. I don't remember how many were involved, but it was fairly large.
Yeah.
Human trial.
It looked really good.
And they said it's not published yet, and to be very careful with the results because the treatment actually killed it.
Oh, that's right. Yeah. Yeah, and several,
Was it a study in the United States?
yeah. Yeah, and some of them had this a cytokine storm response that almost killed them. So the side effects, when you get the T cells going nuts like that, you can have big problems. And so some of the people that gotten really, really sick survived and then didn't have the cancer anymore. But yeah, but 2 people had died.
But this was done on people who only had like 5 or 6 months left to live.
No, weeks. These people, these were dead men walking because they didn't think that this was going to work, and they had complete remission. So that was what was so stunning. So then one of the questions is, can you knock down the dose so that they're not so bad results for somebody who's not so far gone? Yeah. So there's some ethics there, right? Yeah.
That's so interesting. We took some of the genomics students went to see Craig Venter, who was one of the 2 leaders in the Human Genome Project.
Well, it's the human genome is Craig Ventner's genome, guys. Yeah, I told you what I had a problem with that one. The first dog sequence was Craig Ventner's dog, but it's not exactly a dog, it's a poodle. I have a problem with that.
He's one of those guys that really is going to be in the history books. Love him or hate him, there are plenty of people on both sides of that line. But so there's approaches to medicine that these kinds of tests are beginning to enable. So we're talking about things like very advanced cancers where we have these radical treatments, and that's how we've always done things, at least in medicine. But now we're talking about the possibility of seeing, especially if we have just data, and being able to maybe have our insurance companies pay for more preventative measures and early diagnostic tests. Where we're getting to the point where Human Genome Project was a $3 or so billion project that took a decade or more, and now we can sequence several genomes in one day with the sequencing technology today. So it's going to be interesting to see how things proceed. If it's up to Venter, we're doing less of that and more of let's prevent it from happening in the first place. If it's up to
yeah, but then there's always the problem if you start going in and changing the genome. There's so the standard now is you could change the genome of affected cells. So like people with cystic fibrosis, you use a little inhaler and give them the good gene in their lung cells or inject it into their pancreas. But that's not going to change the genetics of anything except those few cells. So they're germline cells. Eggs and sperm are not going to be affected. And so the line now is you don't change germline.
Well, except for in England now, because they just gave them permission to do it.
But then they have to get rid of the embryos after about a few days. So, yeah. 7 days? Yeah. But that seems to be the ethical line, is that we don't change the germ line of humans.
Today.
Well, but I mean, the problem is, like, it's like when they cloned Dolly, you know, we had, oh, that's wonderful, but you don't see the 300 dead and deformed baby sheep that it took to get there.
And your DNA remembers its age, so when they cloned the new animal,
Right.
Its
Yeah.
DNA was old. So you're.
Yeah. So, I mean, the whole unintended consequences starts having kind of multiplying effects when you start talking about dealing with germline cells.
I want to say something to respond to the question of this gentleman. Also, there is something that we have to do. I guess this is a study that distribution of different cancers are different in different countries.
Yes.
Lifestyle, environment, and nutrition. This, for example, in the three states, you see much more colorectal cancer, bronchial cancers, pancreas cancers. This is due to lack of movement, too much meat, hormone-treated meats that they eat,
Maybe.
and alcohol. So if there is no history of cancer in the family, it is very, very good idea to begin at the age of 50 to have more moving, it's not a gym or buying a, say, treadmill at home, reducing the weight, and avoiding alcohol and fat, nutrition. These are things, but if somebody has the trace of cancer in his history or in distance, it is a very good idea to have every 6 months or once a year examination. So just this cancer.
I'm thinking that waiting till 50 is too late.
Yeah, some
A lot of, if you're talking about chemical damage, it accumulates.
For example, somebody who's a heavy smoker, is already cancer chance to get it.
Right, and so your immune system is constantly catching these mutations, or the cells, but some of those mutations just are latent for a really long time until you get another mutation in just exactly the right place.
For example, they have
So-
a healthy life, they never smoked, but at the age of 70, suddenly they got hemophagocytic leukemia.
Right. I mean, wasn't there a study in the last 6 months or so that when they did the statistics and looked at it, it looked like about 50% of the cancers were due just to bad luck from cosmic radiation, and not genetic, not lifestyle, but just bad luck.
In the 40s and 50s, many people worked at the ship harbor, and there was a lot of asbestos.
Right, and those are obvious. But if you don't have any obvious exposures, I mean, we know that being overweight is a risk factor. It's not clear exactly why.
Alcohol and overweight.
Yeah, but it's not clear what the mechanism is and how that causes the cancer.
But it is the percentage of cancer this type of people is higher.
Right, but I don't think losing weight after 50 is going to help you've already done the damage. Yeah. Okay. It's been fun, guys.
Well, thank you, Dr. Harless.
Thank you.